And
Just
just please wait. Please wait. Yes.
Sir, now live, sir.
Professor Lee?
Yes.
Yeah, you may start now. Yes.
Okay. So hello everyone. So I'm Jeong-Hwan Lee at Seoul University, and it's my great pleasure to introduce Professor Kyubong Jo. So from Seoul University. So Professor Jo got his bachelor in the Department of Chemistry Education of Seoul National University in 1993. MS in the Department of Chemistry at the Biochemistry Division at Seoul National University in 1995. And until 2001, he worked for the Samsung Advanced Institute of Technology. Then he moved to the University of Wisconsin to get his PhD. So he got his PhD at Analytical Chemistry Division in 2006, and after that he moved to University of Illinois, and he did his PhD. He got his postdoc training over there from 2006 to 2008, and actually that's the time I met Professor Jo in US, and then. After that, he became a faculty member of the Department of Chemistry and the Interdisciplinary Program of Integrated Biotechnology in 2008. So he's working on the development of microscopic sequencing approaches to visualize sequence information of large DNA molecules. So today he'll be talking about single molecule DNA visualization. So let's greet Professor Jo for his talk.
Hey, thank you. Thank you for your kind introductions, and thank you again for the Professor John Bosco, for the giving me opportunity to present my own research, over the world. Okay, so it's so far away from here, though. Okay, so anyhow, I'm sorry to change the subject, because at the time I submitted my abstract, I didn't realize that it's going to be an hour the presentation. So I just expect 15 minutes. So that is the reason I choose one of the topic. But okay, so it's not 1 hour. So I have to present more general topic of my research. That's the title is a single molecule DNA visualization. The background image is my campus, the Seoul University, and we are in the downtown of Seoul. So that's one of the pictures, and then okay, next one. So more specifically, this is a map of Seoul, and if you visit Korea before, maybe you use Incheon International Airport in one of the island, and then you can travel from here to the center of Seoul by this highway or some train. And at the same time, and then our school is located in the middle of here. And then this is in the Seoul has a highway system outside of the Seoul. That's kind of boundary of the Seoul. And then and this picture I just took in the last week, and that is the buildings located in this small island. Okay, so anyhow, that's the Seoul. And then another thing, I guess you are very familiar with all the map of the Northeast Asia. I'm not sure. And Professor Suri came from the northern part of the India. That was surprising though. So and then I am here. Our Seoul is located in here, and we are in the kind of a center of the Northeast Asia. So we are very close to the Tokyo. Yesterday the Olympic was closed, right? And then that was 2020 Olympic, though it is held 2021, and it closed yesterday. And then next Olympic, Winter Olympic, will be held in Beijing in the January 2022. So that's the another place. And then Seoul is also very close to Shanghai, the big city in China, and also in the city of the Russia is Vladivostok. Okay, so all the big cities are just located. This flight distance of around 2 hours from here to here, the 2.5 hours, and that is 2 hours, and that is less than 2 hours, and this is about 2 hours. Okay, so and that's the position I am here. Okay. Anyhow, so this is the picture I took January 2020 in the SASTRA University, and that's maybe the last time we can travel freely without the fear of the COVID, right? So and then at that time I thought that picture was so interesting, so that is the reason I just put it here, and then because of the COVID, we cannot travel a lot. However, there is some positive side of the COVID. One of them is all the people, not just the scientists, are familiar with the PCR testing, right? So in these days, everyone knows what is the PCR, and this is a real instrument we are using. And at the same time, Professor Suri talked about lots of new biosensor technology, and then that is another great opportunity for us to, test our developed product, right? So, and this is one of the famous one, the Abbott ID now. It is kind of a PCR testing, isothermal amplification method, and DNA testing, but it can detect 15 minutes, right? That is so impressive. And then another one is BioFire and Shapid, and there is another very fast technologies. And then Professor Suri talked about this one, and this is a Korean product like Humasis, and then that is another antigen antibody detector. And I think this is so fascinating. This is a real data. And last week, one of my students told me she had a fever, so I asked her to go to get the PCR test that the government is holding, and then also, but you have to wait 1 day before. During that time, go to the pharmacy store and then get a rapid kit, and then get the test, and then send me the result. And then she tested. Unfortunately, she didn't got any COVID. And then next day, the PCR test also negative. So I think this is the rapid kit is so useful for us. At the same time, the COVID-19 introduced a new world of a vaccine, right? So, nowadays, many people are very familiar with the AstraZeneca and Pfizer and Moderna and Novavax. So, and especially I'm interested in the mRNA vaccine like Pfizer and the Moderna vaccines. And then, so I guess many of you, Professor Lee got the vaccine last week, right? So he got the Pfizer vaccine. So anyhow, the good thing is before the COVID-19, people developed mRNA vaccine for the last 20 years. However, they got any FDA approval because of the toxicity of other possible danger. However, for the last 2 years, last years, we have the huge amount of testing of mRNA vaccine, and I believe that they are the people that are really developing this mRNA vaccine. I guess some of the people in this picture will receive the Nobel Prize 2 months later. I hope so. So, and he is the father of the mRNA vaccine. His name is Drew Weissman. And then she's a mother of the mRNA vaccine. That's not the creation. I just copy from some of the articles. And then her name is Kathleen and Kariko. And then these two persons are the founder of the Moderna. And then these two persons are founder of the BioNTech, and that's the Pfizer vaccine. And then he's a professor of the Langer in the MIT. Anyhow, that's kind of impressive. Okay, so and then I just took I just studied a little bit about what they really did, like Weissman and Kariko, and then that was kind of very interesting. And then this is their the mRNA the mRNA vaccine system, but instead of using the uridine, they used M-methyl pseudouridine. And in that case, they can escape the innate immunity in our body. So that is the reason that the vaccines is really working. I really believe that their achievement is so important. And then recently, one of the company, the name is CureVac in the Germany, and they used the uridine instead of methyl pseudouridine, and they made a new vaccine, but that was not successful. The vaccine, the prevention rate is just like 45%, comparing to the like 95% of the Pfizer and Moderna. So, and I really believe that these two scientists will receive the Nobel Prize maybe in the physiology of medicine in October. I really believe that. And another very important advancement for the like COVID prevention, that is lipid nanoparticles, and that was developed by the Pieter Collis. He is a professor in the University of British Columbia in Canada. And then I think this idea is so fascinating. This is another reasons we can really use the like corona the mRNA vaccine. So anyhow, I also believe that he will receive the Nobel Prize maybe this year or next year. Anyhow, that's the kind of introduction of some of the like a common topic. That's not my research though. Okay, so and then the post the COVID, like what really changed the the words, what does the COVID-19 change the world? I really believe that one of them is like this conference, right? So we are very familiar with like Zoom style meetings, and people that I never heard of Zoom. I just know the like word of Zoom, but that's a magnification, right? But instead of the magnification, nowadays, we are more like, connected by the internet. So that's a big changes. Another thing is, biosensors like us, like we developed a new biosensor. That's the like a testing field for the, real world. And then another thing is like, mRNA vaccine. Now it can be, such a powerful tool for not just for the corona. I really believe that their product will cure the, like a cancer and other diseases. I wish that like one of their approaches is to cure the aging problems. So maybe we can go back to like 20 years something or 30 something years old. Okay. So now I'm going to start my own research topics. Today's webinar's topic is the single molecule DNA visualization. My research usually is about like visualizing DNA molecule, and in this case, we use the very specific the chemicals that is stain the DNA. Representing the sequence information is shown in this figure, and from this profile we can get what kind of sequences are in this DNA molecule. That is like I want to call this one is microscopic DNA sequencing. Okay, so and my topic will be like categorized in the four things like DNA binding fluorescence proteins and golden nanoparticle and sequence specific DNA binding chemicals and proteins. Okay, so the first one beforehand, I want to introduce what is the DNA analysis. I guess you many of you are developing the biosensor. You are very familiar with like DNA analysis. Basically, for the COVID analysis, we are using the PCR and the DNA chip and the next generation sequencing and karyotyping. This is a karyotyping. I guess most of you are very familiar with this kind of terms. Okay. So, and then at the same time, and this is next generation sequencing, and then and there have been many companies competing each other to develop the new sequencing technology. The first one is 454 and Illumina, Ion Torrent. They are the kind of winner. They are still working. And then Helicos is unfortunately it's gone, but that was they used the single molecules. And then Pacific Bioscience, they now they get some attentions because of their uses, and Oxford Nanopore is also very useful. Our lab also has Oxford Nanopore. So this is called the next generation sequencing. And then, but the real winner is the Illumina Solexa machines, and then that was developed by the Shankar Balasubramanian. He's an Indian, but he's a professor in the Cambridge. And another one is David Kloerman. They initiate the company Solexa. Now is the Illumina is merged, and then they are the the company for the NGS machines. So usually I usually check the Illumina stock prices, and then recently I bought some of their stocks. Okay, so and then this is so amazing. Like 2003, their stock price is less than $1, and then right now they are over 500. Wow! So that's 500 times more. So that was so amazing, and that really represents that what is the meaning of sequencing in these days. Okay, so sequencing is so powerful, and then we can use the sequencing so easily, and there is many the facilities around, and then we can just give them a sample, and we got the result. However, these sequencing technology usually limited to the less than 1 kb, and comparing to that, the karyotyping is so powerful. However, the limitation of karyotyping, the resolution is a 10 megabase pair. That's kind of too large. So I am interested in the analysis of the DNA molecule. The size is larger than 1 kb, lesser than 10 megabase pair. So in between of these two technologies. So why we are interested in that size of DNA molecule? Because that large, I want to say that that DNA is a large DNA molecule, and then that large DNA molecule contains more information than just the sequences. Okay, the sequencing just to read the sequences. However, in the DNA, there is a lot of protein binding and epigenetic modification and the structural changes. Also, the DNA contains accumulated DNA damages. That is also important for the transcription and regulation. Okay, so and then and so we want to analyze this one. For that purpose, we have to know what kind of sequences are in this large DNA molecule. So, and in these days, people actually analyzing that kind of things using the technologies, which is called ChIP-6 and 10x genomics and others. So basically, ChIP-6 is started 2007, and then there is a numerous something SIG technologies that is, they just added a protein bind, and then or some epigenetic changes and break down to the small fragment, and then using the Illumina, they read the sequences and then they got some results. Okay, that's the basic idea of ChIP-6. And the 10x genomics is also very popular. That is started 2016, 5 years ago. And then their technology is so powerful to get the information in the single cell genomics. However, these technologies, basically, they are based on the Illumina sequencing machines. So because of that, these NGS technology, NGS-related platforms requires DNA fragmentation. In that case, lots of information will be lost during this sample preparation. So there is the reasons I want to analyze the large DNA molecule without DNA fragmentation. Okay, so in other words, intact biochemical information, we want to analyze these things from this DNA molecule in the nucleus. So this is a cartoons, like a model, and this is actual SEM images. We can really see the DNA molecules like this. That is amazing, right? So anyhow, however, the DNA molecule is so condensed, so it's really hard to identify what kind of thing is happening inside of this nucleus. So therefore, we need a technology to elongate the DNA molecule. So this is one of the movies we took, the large DNA molecule is loaded into this pocket, and after that, we enhanced, increase the electric field, and then DNA molecule is going to the nano channel. Okay, this is so fascinating. I really believe that. And then another thing is this is a single tethered DNA molecule. One end is tethered on the surface, and then using the fluor, we elongate the DNA molecule as shown in this figure. Okay, so but there is two problems, I guess. The problem is, the first problem we want to solve, that is, there is a beautiful large DNA molecule. However, we do not know what kind of sequences is in here. I mean, we know the full sequence, but this is definitely the fragment of the DNA. So, and what sequences are there? Without that, how can we interpret some of the epigenetics and protein binding and others, right? So is it really possible? I mean, the current sequencing technology cannot be applied for this purpose. And another problem is, normally we are using the yo-yo-1 dyes, the chemical dyes, the yo-yo-1. And so, I mean, there are many of them like DAPI, or it's ethylene bromide, and some others. But however, the yo-yo-1 is the most popular because it has lots of advantages like high contrast and really bright colors. However, they have two critical disadvantages. One is overstretching and the photocleavage. So that's the another challenge. We want to solve these problems. So first problem is overstretching. This is a T4 DNA molecule. The size is like that, and then in that, from the crystallographic data, we can expect that that's the expected length. However, T4 DNA is much longer. So that is analyzed very well in this NAR paper 10 years ago. In this case, DNA molecule is melted like that. See, look at that. This DNA is connected. However, there isn't any DNA, there isn't any yo-yo color because the DNA is melted, like make the single strand. Okay, so and then we expect the DNA after the staining, it should be like a B form of DNA. However, it's not really B form of DNA. It's like a melted DNA. In that case, enzymes cannot recognize properly of the specific sequence. That's one of the issue. And another issue of the overstretching is it causes of the photo-induced DNA cleavage. Because of the single strand is so fragile compared to double strand. So, and this is one of the movies we took, this is a little bit faster, like a 4 times playback. And we simply watch the DNA molecule, the tethered DNA molecule, but many of them just disappeared because of the photocleavage. That is a serious issues, well-known serious issues. So we try to solve this problem. The answer is we want to develop using the fluorescence protein and DNA binding protein. So, and with the DNA binding fluorescence protein. So this is one of the examples. The long DNA molecule is fully stained by this simple protein. And then in that case, there isn't any intercalation, there isn't any overstretching, and then that the binding is reversible, and then we can see the the DNA backbone. Okay, so and this is one of the more detailed information. In this case, we named it as FPDBP, which is called fluorescence protein fused with the DNA binding peptide. So this is a peptide, very simple peptide, based on the KWK. And then we made 2 colors. One is a green, that's EGFP, and then another one is M-cherry, that's a red color. And then DNA molecule is stained using this simple the protein. So, and the idea we got from one of our previous work. In this work, we used the neutral-Avidin coated surface for a single molecule tethering using the biotin and avidin, the chemistry. However, we realized that the DNA molecule is often bind on the DNA the neutral-Avidin surfaces without biotin. So, and in the beginning, that was a huge problem. And later we figure out why that kind of thing happened. And we analyzed their structures. And interestingly, there is a tryptophan and there is a lysine, that's a WK. And then just before that, there is a DND, that is aspartate. So if the magnesium is attached here, the binding is much stronger. And then we're looking for some of the papers, and then long times ago, 1974, the Helens groups and analyzed KWK for the DNA binding, and KWK is a common motif in the many of the DNA binding proteins. So we thought we decided to use the KWK for the DNA binding peptide. So this is one of the example, and this is a EGFP in the middle, and then KWK in both sides. And then this is M-cherry in the middle. And then this is a yo-yo-1 stained DNA molecule. So this is more similar to the yo-yo-1 staining. However, without the chemicals, the organic chemicals, we simply use the binding, the DNA binding proteins. And then this one is so advantageous because it completely solved the phototoxicity. This is one of the example. Yo-yo-1 stained DNA is so bright in the beginning, but it has the breakage here and there, and after 30 minutes, they are fragmented. It is fragmented into small fragments, right? But in this case, EGFP staining DNA molecule without any beta-marker pattern, it survived, not just the 30 seconds, 5 minutes and 10 minutes. It is stable, very stable. Okay, so that we solved the phototoxicity problem. And another advantage is another problem we solved using this this FPDBP, that is overstretching. So this is a yo-yo-1 stretched DNA molecule, and this is the FPDBP stained DNA molecule that is pretty much the same of the natural DNA molecule itself. Okay, and then in addition, we used this one for the the staining cells. This is a bacteria. And then compared to in the control experiment, we were able to see the DNA localization in some spot of the bacteria. And then this is the human cell, the HEK293T cells. And then we can see the nucleus. And then inside of the nucleus, there are some aggregated part for the DNA molecules. So we believe that that is the visualization of DNA inside of the cell. Okay, next one, I talked with Professor Lee in one of the in the pub, and we drank a lot, and then we thought, okay, what about just adding the golden nanoparticle instead of the fluorescence protein? This is one of the images, and the golden nanoparticle is aligned along the DNA backbones as shown in this figure. So, and the idea is more like this. We just add a SH group after the a KWK, and then we add golden nanoparticles, and then now the golden nanoparticle has this DNA binding peptide, and we just added this nanoparticle on the DNA backbone. After that, we grow the golden nanowire. So this is one of the example of golden nanowire grow on the DNA backbone. Okay. So, and this is the longest DNA molecule we have observed, using this method. And this is common way of the staining DNA using the yo-yo, and then on the fluorescence microscope. That's the length, and then we cannot see the details. However, this is the nanowire looks much longer because we use the electron microscope to visualize this DNA molecule. Okay, next one, not just the stained DNA molecule. We thought, that there are many of the DNA binding protein has the capability to recognize specific DNA sequence. I thought it is possible to stain the DNA molecule in the sequence specific way. So this is one of the example. So, and the first thing, we added various colors, many of the, fluorescence protein is added to the DNA binding motif. And then we were successfully able to visualize the individual DNA molecules using different FPDBP. And then also we changed it a DNA binding motif. In the case, not just the KWK, we used HMG, HNS, BRCA1, and many others actually. So, and then one of the data showed very interesting result, and that has some fluctuation that is represented like a AT specific pattern. So using the HNS M-cherry, we simply reduced the concentration, and then we were able to visualize the AT-specific region using this one. Okay, after that, to compensate this, unstained region, we add like a BRCA1 EGFP. This is one of the results. So now we use the two colors and then to get some sequence-specific information. And this two-color staining is so advantageous because these molecules, now we can tell the direction of the molecules based on these patterns. And then there are some of the fragmented molecules, and this fragmented molecule is exactly the matching of this one. And then this fragment is matching of this one. So I think this is so powerful. Also, we can use this one as one of the kind of a sensor type platform. And then in this case, these two molecules like 7 kb and 8 kb, their difference is just 1 kb. So in other words, the sizes are similar to each other. However, the AT patterns are not the same. So in this case, it's much, the reddish color comparing to that one. So using that, we can identify which one is which based on this one. So, and then after I published this one in the paper, and then I met another scientist from Germany, and then she asked me, is this your FP-DBP working in the PBS? And then because PBS is common, phosphate buffer cell line is a common buffering system to mimic the physiological condition. So, but most of the experiment that I presented so far, that is just like a 1XTE, that is just like 10 millimolar of the trees and 1 millimolar of EDTA. So I just checked what is the the concentration of PBS that has a huge amount of sodium chloride and a little bit of the potassium and then another sodium. So just comparing to that, that is about like 150 millimolar of the sodium or potassium ions. That is too much. So, but we try to use our FP-DBP for the PBS, but in most cases, it's not really working. So, and I thought maybe the answer is using some of the DNA binding protein working in the inside of the nucleus, because the nucleus has actually the more than the PBS does salt. Okay, so I thought we can use the tail. Tail is the transcription activated ligand factor, and that is known to recognize the specific sequence inside of the nucleus. So, and also it has the sequence specificity. So, and also we just checked the tail can be linked to the fluorescence protein, and there is an example of the tau effector is fused with the fluorescence protein. So okay, I thought, oh yeah, it's possible. And then also the inside of the nucleus, they stain the DNA molecule using the tail FP. So we tried to do that. So but usually the tail has 20 recognition sites. That's too many. So we just reduced the size of the tail, and that is much more efficient for the cloning though. And then the 7-mer of the the tail the tail. So we called it a truncated tail FP, and they can recognize the multiple sites in the bacteriophage lambda. Interestingly, instead of the sequence specific pattern, they just stain the whole DNA like other FP-DBP. Okay, that's the one thing. The question is it working in the 1X PBS? So this is the 1XTE, it's working so well, but in the 1X PBS, no, we cannot see anything. Okay, that's a little bit disappointing. But my students came to me, and then, she just tested in between of the 1XTE and the 1X PBS. That there is she simply increased the sodium chloride concentration. And then that shows some of the patterns. It looks like AT-specific patterns we saw in the previous studies. Okay, so, and then we tried to understand why that kind of thing happened, and then it is our result using the fluorescence measurement, and then this frat has much more specificity to 80 specific reason compared to the juice-rich reason in the like 100 millimolar or 50 millimolar and 70 millimolar. So that is the reason. This one has like 80 specific pattern, although they have a much higher specific sequence specificity about like 200 millimolar of sodium chloride. Okay, so that's really useful. So we started using this one. Previously, we used the yo-yo one, and then we want to visualize the DNA digestion using the yo-yo one stained DNA molecule. However, when we loaded like EcoRI one, the DNA molecule is simply disappears. That is like a photocleavage. Compared to that, we use the tail FP, truncated tail FP, and this is DNA molecule, and then we added the EcoRI one, and then there is some digestion, but the digestion is not random. It is they just cut at the expected position, like in this figure. So we just like monitoring the one of the molecule to which side is digested. Okay, so I think this is a very useful tool to monitoring the like restriction enzyme digestion. And then after that, we added like some of the like sodium chloride, and then each DNA molecule shows the 80 specific pattern. Based on this 80 specific pattern, we can confirm that digestion is really the position we expected. So, and this is one of the map of the X bar one should cut in here, and then that is the 80 specific pattern, and this is the actual molecule we analyze using these technologies. Okay, the last topic I'm going to talk about, that is the sequence-specific DNA binding chemical. So that for that purpose, I work with Hiroshi Sugiyama. He's a professor in the Kyoto University. The first picture I presented in this today's talk. So, and at the same time, our lab developed various kinds of the DNA binding protein and sequence-specific binding protein. But at the same time, we found one of the paper from one of the Sweden's group, the Westerlund, Frederick Westerlund, and they used the netropsin that has the 80 specificity and yo-yo. So because netropsin is occupied the 80 specific region, yo-yo cannot bind the 80 specific region. So that is the reason they generate some patterns that has a sequence specificity shown in this figure. Okay, so and I thought maybe, but instead of using the netropsin, what if netropsin linked to a dye? Unfortunately, I'm not an organic chemist. I do not have, I know what is this chemical, but I don't know how to synthesize it. That was a problem. And then this is a netropsin. And then is it really possible to connect this netropsin with any dye molecules? If it's possible, we can show the 80 specific pattern like this, right? In the inverse of the Sweden group's result. So, and we look through the literatures, and similar to the netropsin, this is a netropsin. It has a 2-pyrrole, that is important. Distamycin is a 3-pyrrole, that's another characteristic. And this kind of pyrrole has the pyrrole molecules has the sequence specificity of the 80 specific regions, especially like in the in the minor group side. And then in the imidazole has an N in here instead comparing to that one, right? The nitrogen in this side, and then imidazole and pyrrole pair, they selectively interact with the GC. That was started from the 1994 in Science, and Peter Derva in the Caltech University. And after that, some of the people are trying to develop the new way of the DNA binding protein, not protein, the polyamide. And then later, I met one of the conference, the Hiroshi Sugiyama. He's the expert of developing the pyrrole imidazole polyamide. So I thought, oh yeah, instead of asking the other organic chemists to synthesize that one, simply I just can work with him, right? So, and I work with him, and then he synthesized these chemicals. We tried multiple times with the various ideas, and then finally we found this chemical 8 pyrrole is located in the linearly, and then this one shows the like a really clean the 80 specific pattern as I shown in the first images. Okay, so and first thing, we always using the lambda as a control experiment. So we use that and we can simply identify which direction they are because they have a very specific patterns. So this is a Tamna polypyrrole. We use the Tamna dyes for the like, the fluorescence dyes in these studies. And this is like their patterns from these images. And this is the 80 frequencies and they exactly to match to each other. So we use this one for the like a lambda concatemer. In that case, a heptamer, the 7 sequences are connected to each other, and we can simply repeat the sequences like that. And then this is pentamer 5 times, and 4 times, and 3 times, and this is monomer. Okay, so this is working so well to identify the each unit for the lambda concatemer. At the same time, we used like chromosome 1 of the S. cerevisiae, the yeast. And then the whole size is 230 kb, and we stained the yeast chromosome with these chemicals. And then we saw some of the specific patterns, then we aligned like these figures. And then we can see we can cover the whole chromosome using these technologies. So next challenge is we want to try to use this one is a little bit bigger, the genomes like EcoRI genome that is 4.6 megabase pairs. And then we obtained EcoRI genome that will be randomly fragmented like this. And then we stained that chemicals and then get the images. And after that, we developed some of the programs to search for the whole genomes and to identify the highest matching point, calculating the cross-correlation values. So this is one of the images, and from that we can just calculate this the radish the profile, and then we match this radish profile with the in silico 80 profile map as shown in this figure. So we just believe that that is originated from here. Oops. So and we were able to complete the EcoRI genome like this, and these are the example of the the multi copies is aligned to the same the DNA fragments. And then this is the largest DNA molecule we obtained for this experiment. The size is 500 kb, the long DNA molecule, right? So, and then because we used EcoRI, the K12 standard EcoRI models, we know the full sequence of this. And then according to the NCBI website, and these are the genes in the inside of this DNA fragments, okay? So, and we simply magnified this, very small regions. We can identify the various kinds of the genes, located in these regions. Okay, in summary, today I present the 2 main topics. The one is the DNA binding fluorescence protein and golden nanoparticle. The first one is they just stain the DNA without any sequence specificity. The second topic is we developed the sequence specific DNA binding chemical and proteins. Okay, this is a chemical, this is a protein. Okay, so and then this is the end, and this is my group, and thank you for your attention.
So thank you very much, Professor Zhou, for the very interesting talk. Please ask any questions if you have any. So I have a couple of questions to ask. So you showed the thing that if you treat the DNA with yo-yo dye, the DNA was cleaved. Is it like the unique property of yo-yo dye, or does it normally happen if you treat DNA with fluorophores?
It depends. So especially dyes, the intercalating the DNA molecule, they have high probability of the photocleavage. So I'm not sure why that kind of thing happens. So people usually use the oxygen scavenging system to prevent that kind of the phenomena. However, that is not very easy. Okay, so normally the DAPI is like attaching to the minor groove, and they are relatively safe. Okay, so but the ethenium bromide, they are intercalator, and that causes like a DNA breakage.
So another question I have is, the technique that you have shown using single molecule imaging is very kind of unique. So it is different from the technique people normally think about. And you mean what is NGS technique? So how different do you think? What kind of so the information that we get will be different from those the information we get from those techniques.
Okay, so also the single molecule people for the like NGS people is also using the single molecule. So there are two groups of the analytes. The first one on the top, like the 454 and Illumina and Ion Torrent, they are based on the amplification, not exactly PCR, however, it's similar to PCR. And they try to reduce the noise information, okay? So, and then, but the single molecule, the bottom three technologies is based on the single molecule. And there are some advantages. I mean, the PCR is not omnipotent, okay? So PCR cannot exceed like, 10 kb or, 50 kb. I mean, sometimes they can do the like, 20 or 30 kb of the DNA, but that's not very efficient. However, if you can read the single DNA molecule, you can get the information of a much larger one. However, the issue is, normally we are using the like polymerases and or some electrochemistry. In that case, there is some noises, and that noises causes some of the error data. So that's not perfect. Mm-hmm. So people try to develop a new one in the to get rid of that kind of noises, but it is not easy. Okay.
Okay.
Yeah. So but another advantages of the single DNA molecule, that is protein binding and epigenetic modification and structural damage changes, and especially like damages. These events is usually they are random single molecule event. Okay. But currently, we just mix them of the multiple cells and get the average result. But that's not accurate. That's not what we want, right? But the single molecule is the only method to really see what kind of thing really happened for the single DNA molecules.
Okay. So, any questions from the audience or from here in the Zoom? So thank you very much for Professor Joe for the really interesting talk. Really appreciate. And then I forgot to mention that you indeed went with me to SASTRA University, and then we enjoyed there very, very much. Hopefully we have time like that later on. Let's see.
In Korea.
In Korea, yeah.
Yeah, in Korea. Thank you for the John.
Okay, thank you very much, Professor Joe. Okay.
Yeah, thank you. Thank you for having me.
Thank you very much, Professor Joe. Just 1 second.